Okay, hello everyone. Today I'm going to be giving an overview on the cell cycle, and I'm going to be talking about, first, I'm going to be talking about the job of G0, or the rest section of the cell cycle, because it is quite simple and I'm not going to be talking about it later. So as you can see here, G0 is in between G1 and the S phase, and in G0, the cell can, if it's a neuron or if it's a muscle cell that doesn't need to replicate, it can just go there and do its job. Or if it doesn't have enough resources, or if it doesn't have, or if it's not healthy enough to replicate, it can also stay there until it is. Next, you will see that I'm drawing what look like green triangles, which mean go, and red stop signs, which mean stop. And these are supposed to signify the cell cycle checkpoints or regulators. Now these are proto-oncogenes, and these are cell cycle inhibitors or tumor suppressor genes. And we will cover these later, but proto-oncogenes, an example of these are, well, they're genes that make proteins like, say, a gas pedal in a car, which would make the cell cycle keep going. An example would be a cyclin, which activates a cyclin-dependent kinase, which I will also cover later, to advance the cell cycle. And tumor suppressor genes, an example of a gene that an example of a protein that this gene can create is P53, which will also act as a brake to contrast the gas, and so it could actually stop or pause the cell cycle and theoretically fix what happened that went wrong. But sometimes the cell is damaged beyond repair and has to initiate apoptosis, or basically kill itself, because it is better for the greater good of the organism. So now I'm going to talk about cell cycle regulators, and specifically cyclins and cyclin-dependent kinases. And as you can see in the graph that I'm drawing here, the cyclins vary in their number in the different stages of the cell cycle, which is good because when they're bonded with a kinase, they go and activate proteins that transcript sections of DNA or genes that will then create the actual proteins that will advance the cell cycle. So if they were always working by themselves and they didn't need a cyclin, that would be a problem, because we would have an issue of the cell cycle not stopping. But there are many other oncogenes that are affected by and mutated much more easily. So those, as well as tumor suppressor genes, have to watch out, because if they were to be mutated, if the gas pedal were to be made to be an oncogene and stuck on and not be able to lift off, or if a P53 gene or P53 protein or a gene for P53 were to be mutated so that it wouldn't be able to produce P53 and not be able to brake the car, that would be also bad, equally bad. This leads to cancer, which is when a cell with a mutation can go through the cell cycle without stopping, even if it has an issue, because there are no cell cycle regulators to see whether or not the cell has made the checkpoints. Another point I'd like to make: there are tumors that are caused by these cell cycle replications that are uncontrolled, and there are also malignant tumors, which are cancerous tumors. The difference being that cancerous tumors can spread throughout the bloodstream to other parts of the body, inhabiting other organs in the body, while benign tumors do not. Okay, so now that I'm through all of that, I'm going to start talking about the G1 phase, which is the first step in the cycle. So in the G1 phase, this is the initial growth of the cell after it just came out from a parent cell. And I'd like to point out that this is what is known as a diploid cell, which is a cell that contains a full set of chromosomes, half from its father and mother, and the other half from the mother. And so basically, all the G1 cell has to do is to grow and then decide whether it's going to go into G0 or not. And this is also related to its checklist summary with the cell cycle regulators, which check to make sure that G1 cells have no DNA damage before going into synthesis and have sufficient resources to engage in the rest of the cell cycle. The synthesis phase is where there become to be sister chromatids, which are first of all, let me define what a chromosome is. A chromosome is condensed genetic material. The synthesis phase is supposed to copy all of that and make it double. So we have an n equals 3. We have an n equals 3 cell here, which I should have mentioned earlier, but that means that there are 3 separate pairs of chromosomes in G1, which means that now there are going to be 6 separate pairs, or 12 chromosomes, and these will be arranged in sister chromatids, which are separate chromosomes joined together by a centromere in the middle, and then once they're joined together, they are also known as a unit, as a chromosome. In the synthesis phase, after this is replicated, the DNA replication, or the replication regulators, or the cell cycle regulators, I should say, check to make sure that there are no errors in DNA replication before advancing to the next phase, which is G2. In this phase, the cell is continuing to mature and grow, and it completes the replication of other organelles and very important components, such as the centrioles, which are the parts of the cell that are very crucial during mitosis. They are the ones that hold the spindle fibers, which are the fibers that will help, kind of like the hands that will help organize the chromosomes so that they can be split evenly to the 2 sides of the cell that will then split apart. So basically the checklist summary of this, and the things that the cell cycle regulators look for in this, are to make sure that the DNA has no damage, the chromosome set is complete, and that there are enough cell components so that there are enough cell components and every other component so that there are enough for 2 separate cells later down the road. Okay, so now we're going to talk about mitosis and the details of mitosis. The first step being prophase. In prophase, as you can see in the bottom circle, which I want to draw more attention to now, up until now, the bottom circle used to be a cell. Now what it is the nuclear envelope, which consists of the membrane that surrounds the nucleus and the nucleolus. Now you can actually see the sister chromatids together. They're visible under a powerful microscope, but before they weren't. If you look back in the video before, they were just they were too uncondensed to be visible. So in prophase, the chromosomes and the sister chromatids are condensed, and the separate centrioles are almost to the opposite sides of the cell so that they can connect to the spindle fibers, which are the protein structures that divide the genetic material, or the chromosomes, later on in mitosis. So we have the centrioles close to the other side with the spindle fibers emerging, and the chromosomes are greatly condensed in this stage. In this stage of the game, after prophase, which is called metaphase, the sister chromatids, or the chromosomes, are aligned down the middle by the spindle fibers and the centrioles, so that this is a very important step leading up to anaphase. Now in anaphase, since there's an even split down the middle of the chromosomes from metaphase, now they split, and so that the centromeres are pulled apart, and the sister chromatids are now known as separate chromosomes, and so that each side of the cell gets 6 equal chromosomes, and at the same time the organelles begin to migrate so that each cell that is starting to split will have an equal amount of everything. Here in telophase, which is the last step, you can see that the cell is really starting to bubble into 2 separate cells. And the splitting into 2 daughter cells is known as cytokinesis. That's the process. And so in telophase, each side has its own centrioles, each side's got its own organelles, enough chromosomes, so they're really starting to split apart, and there's separate nuclear envelopes, so each side is really starting to separate from each other, and this is the last stage before they are 2 independent cells. The last thing I'd like to point out is how in this stage in the game, through mitosis, the cell cycle regulators are checking all along to make sure that the chromosomes are attached correctly to the mitotic spindle to avoid for devastating errors during mitosis.